Hello, I'm Janine with Natural Grocers, and I'm very pleased to introduce Barbara Swanson. Barbara is a nutritional consultant with a focus on whole foods and superfood-based supplementation based on the tenets of functional nutrition. Her award-winning book, Beyond Foods, The Handbook of Functional Nutrition, offers a unique health model to explain the very complex subject of how nutrition creates health. In this presentation, she will be discussing the truth about GMOs. Welcome, Barbara.
Thank you, and thanks everybody for being here. Let me get my presentation up. And yes, today I want to talk about why you might want to say no to GMOs. So GMOs are safe. Wait, no, they're not. So what do you believe? Because you will hear equally fervently both sides of that argument. It's important to understand the world is filled with misinformation, way more than a single person could absorb in 100 lifetimes, literally. The problem is, in our digital and social media world, information is not always what it appears to be. There are often opinions which masquerade as facts. There are facts that are taken out of context so that they appear to support a fiction. And then there are outright lies and misinformation, which are meant to either reassure you or make you so confused that you just give up trying to find any actual truth. This presentation is to present you with some basic facts so that you can make an informed choice on whether you want to use GE, genetically engineered foods. And in this presentation, I use GMO, which stands for a genetically modified organism, and GE, which is genetically engineered, pretty much interchangeably, as they pretty much mean the same thing. So a few starter facts. Humans have been practicing agriculture for over 10,000 years. And actually, if you really look into it, it's probably closer to 18,000 years, but 10,000 years is the accepted timeline given. Another fact is that the commercial use of genetic engineering in crops is only a bit more than 25 years old. It began with the Flavor Saver tomato, which went on sale in the spring of 1994. The purpose of that tomato, by the way, was to stay good longer, not to taste better, not to be more nutrient packed, not to increase production, just to ship better so that more money could be made. Another fact is that genetically engineered crop production has skyrocketed. Nearly 90 times more land is under genetically engineered production than just 20 years ago. Yet across the planet in 2017, genetically engineered crops were farmed in only 27 countries. Only 5 of the 39 developed countries allow genetically engineered crops in their country. 41% of all the acreage worldwide that are planted with GMO crops are in the United States. So why are we one of the only 5 developed countries to allow genetically engineered foods, and why do we produce so much of it? Money. So how were GMOs determined to be safe? Before genetically engineered foods hit the market, and even factory livestock farms, which began in the 80s, they started feeding livestock genetically engineered corn. 2 things occurred. The presidential administration at that time decided that biotech foods did not present any new risks to the consumer, so no new laws were needed to test GMO foods. In other words, there was no law that said you need to test this for its genetic safety. At the same time, the FDA decided that since the final genetically engineered product did not, quote unquote, vary significantly from the original, they didn't need to regulate them. Now, this is important because remember, we have something like 97% of the same DNA as an earthworm. So saying something does not vary significantly from the original is a pretty wide variance of what it actually produces. So what that happens is that this leaves genetically engineered manufacturers the power to label any product they produce as GRAS or generally regarded as safe. And this is the standard by which they can sell in the marketplace. So how can they do this? Because GRAS determination is decided by the manufacturer that created the ingredient or product. It is never decided by an outside authority, which is a blatant conflict of interest and has been an issue for 40 years. There is no obligation on the part of a manufacturer to conduct long-term safety studies to determine GRAS certification. And since seed companies actually patent their genetically engineered seeds, researchers have little or no access to those seeds to do any kind of independent safety testing. They're not allowed to have them. So third-party studies on GMO crops is pretty much cut off at the get-go by the manufacturers owning those seeds. So what about the safety of GMO food? So one of the main support points for the safety of GMO crops is that the FDA allows them. We're told that the FDA did research and discovered no potential issues at all, and that's why they were allowed to be sold. But the truth is, FDA scientists have repeatedly warned that genetically modified foods can create unpredictable, hard-to-detect side effects, including allergies, new toxins, new diseases, and potentially nutritional problems. They urged long-term safety studies, but they were 100% ignored. So, is scientific truth being suppressed, misleaded, rejected, denied in the name of progress? There's some good evidence that at least in part is happening. And I give some qualifiers here. I'm a fairly good researcher. It's really hard to find the truth, if there is a simple truth, when it comes to GE and GMO. There are plenty of websites assuring you of the safety of GE foods. 100% of those sites are supported by the industries that produce the seeds or the poisons that the seeds are created with. There are plenty of websites talking about how unsafe GE foods are. 100% of them are typically not necessarily very scientifically based as far as what they can give me for their science behind it. But there is some definite science around why GMOs are not safe. But first, before I get into that, I want to talk about how GMOs are even created. So there are currently two main methods using to produce a genetically modified organism. Each rely on changing only specific genes within your chromosome. So let's look at what a gene is, because I think it's pretty darn confusing. If you take a look at the top of this schematic, you'll see there's a cell with a nucleus, and inside the nucleus is your chromosome. Now, inside your chromosome, there's what they call legs, and surrounding each leg is what's called solenoid loops. And you take a look at that, and this is basically like an insulating binding. And that insulating binding is making up of things called nucleosomes and histones. These are ways that your body will turn on and off genes within your chromosome. And then inside the histones are attached to the outside of your actual DNA. And a gene will be several loops of your DNA with several base pairs of your chromosomes inside that. So, or excuse me, the base pairs inside your DNA. So a gene is several loops of these pairs. So it's very complex. A gene is not a single thing. It's a complex of many different pieces of your DNA. That's what I'm trying to say. It's not a small individual thing. It's many components, all of which affect one another in ways that can then also be affected by your environment and what you eat. So let's take a look at how you actually create a GMO, knowing it's going to target a gene. There's two ways. One of the ways goes off to your left and the other way goes off to your right. So on your left-hand side, you have what's called an agrobacterium. This is a bacteria. And what happens is the scientists find a gene that they think will work for some reason. One of the best examples is the fish gene in your tomato. They found a gene in fish that keep it from freezing. So they thought, well, what if we put this gene from a fish into your tomato? It won't freeze, or it will help maintain the freshness of your tomato so we can sell it for a longer period of time. So they took the gene out of the fish and they would insert it into the bacteria. The bacteria would then grow that gene. And then your bacteria is mixed with plant cells. And the plasmid, which is a type of biological thing, moves into the plant cell and inserts that fish DNA into the plant DNA. Now, how does it insert it? Why does it insert it? What does it do when it inserts it? We have no answers for any of that, by the way. We don't know those answers. And at that point, then the new plant cells are screened and they get rid of the ones that didn't get transferred by the bacterium, and they only keep the ones that were. The other way to do genetic modification on the right-hand side is called a gene gun. And what happens is the gun is loaded with a particular biological construct, which I will talk about in a moment. That construct is then added to gold particles or titanium particles that have been coated with DNA they want to insert into a plant. And then they use a gun and they shoot those particles into plant cells where they will randomly access and change the DNA that they come in contact with. And again, they search through the plant cells till they find the one that they think is the right one, and they keep that. And now you have a transgenic plant with a new type of gene in it. So let's take a look at your gene gun, because when I first read this, I actually thought it was a joke. I thought, surely science is better than this, but it's not. So basically, there's a gene gun barrel, and then there's a plastic disc, and on that disc, it's coated with gold particles that have been covered in the DNA that they want to get into the plant cell. And then there's a screen, and the gene gun shoots a specific biologic substance through this screen, carrying this DNA covered gold into the target plant cells, where that can then change the DNA. And I'll show you how that happens. It's using something called the Crispr technique. What it is that they create a genetic sequence. Scientists create this. It's not something found in nature, called a guide RNA. It's as close as a match as they can get to the DNA that they want to replace with something new. So that guide sequence is then mated to an enzyme that will cut out what's in the plant and then dissolve. And then the DNA that was injected with gold will take its place. And now they have a brand new gene. Now, that sounds as random to you as it does to me? Because to me, this sounds totally random. I'm assuming there must be more to it than I understand. I don't want to get into all the science, but that's pretty astonishing to me. Those are the only two ways that DNA is modified currently. So safety. We can right now be fairly assured that acute issues like dropping dead after eating GMO corn, that's not really happening. Because GMO corn is everywhere. If you're not eating organic corn in the United States, you are eating GMO corn. 99.9% of the time, if you're not eating organic corn in the United States, you are eating genetically modified corn. So, if we were going to start dropping dead from this, that would be happening. And it's not. But there is a very real concern with what are the chronic long-term side effects of using genetically engineered crops. This is a great picture. This is genetically modified rice. And you'll notice that they're wearing containment suits because they have no guarantee of the safety of this rice, not even its dust. I find that pretty interesting. I think it's important to note that one of the main reasons genetic modification occurs is to supposedly increase crop production by requiring less pesticide use. What has happened is that, and the reason Monsanto is involved, is that a great deal of genetic modification involves inserting pesticides into the genetic structure of a seed so that that seed cannot germinate without the addition of more pesticides. That's a literal fact. Now, it supposedly requires less pesticides as it's growing, but the pesticide is inside the corn, and specifically in America, it's corn that's done. So long-term issues, they happen because our genetic expression is dependent on the action of histones. And I talked about histones. They're on the outside of your DNA in that solenoid loop, and they actually are an integral part of changing how a gene will act and interact in life. They turn genes off, they turn genetic expressions on, and your histones are affected not only by the gene itself, but by the outside environment. Well, when you change a gene, you're going to change the histone interaction. So potentially eating genetically modified organisms can also affect our histones and thus our DNA expression, because we're not eating just the changed gene in the plant, but also the genetic chromosomal information that affects how we interact with our world. And that affects our epigenetics, or how we interact with our environment and how our health interacts with our environment. So in the Center for Food Safety, one of their concerns is immunosuppression. The well-respected British medical journal, The Lancet, published an important study which examined the effect on rats of the consumption of potatoes genetically engineered to contain the biopesticide Bt. Scientists found that rats consuming genetically altered potatoes showed significant detrimental effects on organ development, body metabolism, and immune function. The biotechnology industry has of yet not produced a single study of their own to refute his findings. Moreover, 22 leading scientists recently declared that animal test results linking genetically engineered foods to immunosuppression are valid. Allergic reactions. The genetic engineering of food creates two separate and serious health risks involving allergenicity. Genetic engineering can transfer allergens from foods to which people know they are allergic to foods they think they are safe. This is not a hypothetical risk. A study by the New England Journal of Medicine showed that when a gene from a Brazil nut was engineered into soybeans, people allergic to nuts had serious reactions to the engineered product. Without labeling, people with known food allergies have no way of avoiding the potentially serious health consequences of eating GE foods, which may contain hidden allergenic material. The second allergy risk associated with GE foods is that they could be creating thousands of different and new allergic responses. Each genetic cassette, which is the gene, it's a cassette, being engineered into foods contains a number of novel proteins. These are altered genes, bacteria, viruses, promoters, marker systems, and vectors that have never, ever been part of the human diet. The FDA was well aware of this new and potentially massive allergenicity problem. Once again, the agency's own scientists had urged long-term testing and were shut down. Antibiotic-resistant bacteria. Virtually all genetically engineered foods contain antibiotic-resistant markers, which help the producers identify whether the new genetic material was actually been transferred into the host food. So they mark, did this genetic transfer work, with an antibiotic-resistant marker, or making that food antibiotic resistant. The large-scale introduction of antibiotic marker genes into food supply could render important antibiotics useless in the future. For example, a genetically engineered maize plant from Novartis includes an ampicillin-resistant gene. A number of European countries, including Britain, refused to permit Novartis Bt corn to be grown due to the health concerns that ampicillin-resistant gene could move from the corn into the bacteria in the food chain, making ampicillin far less effective in fighting a wide range of bacterial infections. Now, could this happen? We don't know. That's the point. We don't know if this can move into the surrounding soil, into the bacterium that this corn is grown in, because no testing has been done. The BMA, the British Medical Association, addressed this problem in its own study of GE foods. The BMA's conclusion was unequivocal. There should be a ban on the use of antibiotic-resistant markers in GM food, as the risk to human health from antibiotic resistance developing in microorganisms is one of the major health public health threats that we will see in the 21st century. And indeed, we are seeing that. So studies on the safety. So the following is from PubMed. The results of most studies with GM foods indicate that they may cause some common toxic effects such as hepatic, pancreatic, renal, or reproductive effects, and may alter the hematological, biochemical, and immunological parameters. In other words, they can affect your liver, your pancreas, your kidneys, and your reproductive organs. And they may change your blood, biochemistry, and immune function. And this is the Department of Forensic Medicine and Toxicology out of Athens. Gut health, pictures are worth 1,000 words. So on the left-hand side, you have a healthy villi in a gut. And on the right, you have the villi after having been exposed to GE corn. And you'll notice at the base of the villi, you'll see these little like dark spots with a white patch inside. Those are where your immune system and your gut live. And if you take a look at the one on the right, you'll see they're vastly expanded. They're diffuse. They no longer look like they have really good separation in their parts. There is a chaoticness to the cellular structure. And you'll notice the villi themselves are almost like melted, which signifies significant gut deterioration. And since your immune system is based in your gut, or at least a good deal of it, this is a significant risk. And then here's another picture of what it looks like when you have eaten genetically modified foods. And again, you'll see that the villi are diffuse. They're no longer separate, like little separate tendrils. They are almost converged completely together. You'll see that the base of it, where it should be your immune function cells, have now traveled up the length of that villi, and it's even more chaotic than before. So you're looking at real-time damage to the gut, introducing genetically modified foods. I believe this study was done on corn. So let's talk about the Bt toxin, which is the main and first use of genetic modification. Bt has been used as a natural insecticide since the 1920s via a topical or on top of the plant application. So Bt toxin insertion into plant DNA was supposed to effectively reduce susceptibility to pests. As they discovered after a lot of work, only 2 crops actually work on pests, corn and cotton. The majority of crops use this technique to make the plant non-susceptible to death by the application of herbicides. In other words, they can now add lots of herbicides to kill pests and it won't kill the plant. That plant can absorb more poison. GMO rodent feeding studies, stomach mucosal barriers compromised, increased intestinal infections, hyperlipidemia, which is fat in the blood, higher blood glucose levels, lower birth weight, increased mortality or death of offspring, disturbances in the pancreas, livers, kidneys, adrenal glands, ovaries, testes, thymus, immune system misfunction, blood biochemistry changes, upset in the functioning of your digestive system. So in summary, GE crops and GMO foods were really meant to offer a promise of more food per acre. And if you read industry publications by the GE industry, they will say that that absolutely is happening. I could not find independent verification of that anywhere, but I also could not find that they were not telling the truth. So I leave that open. That's what they say they're offering. I would say, considering the changing climate and the increasing demand for what are already scarce resources, it is my opinion that expanding any kind of industrialized agriculture is not a great long-term solution. Agriculture is already one of our largest contributors to climate change. In order to sustainably produce more food, a transition to more regenerative farming methods makes sense to me. And this actually has a name. It's called agroecology. It's the application of ecological science to the study, design, and management of sustainable ecosystems. They are based on local ecosystems. It has the potential to enhance an agricultural system by creating beneficial biological interactions and synergies among the components of that specific agroecosystem. Our world is varied and different. One solution is not going to work everywhere in the world. We need to get back to local care of local resources, especially when it comes to agriculture, in my personal opinion. Thank you for watching. If you would like a free in-depth health report, you can go to yourguttype.com/health review.
Thank you everyone for watching. Remember to like the video and subscribe for more virtual classes from Natural Grocers. Are you looking for personalized nutrition advice to support your unique health goals? Please visit naturalgrocers.com/virtual-coaching to book a free one-on-one nutritional health coaching session with your nutritional health coach today.